Go. Welcome back. Now let us see emasculation, cross pollination, and self pollination in case of cotton. As these crops are dwarf and the season is Rabi season, so the height of the plant is small and it is dwarf variety. So first start with the floral biology of the plant. So this is one of the flower. The scientific name of cotton is Gossypium species, as there are different species in cotton and the chromosome number also varies according to the species. So let us see about the floral biology. Sepals are 5, which are, so these are the 5 sepals. Petals are also 5, which are polypetalous, which means they are free. They are not fused together. See, there are 5 petals. And it contains numerous number of stamens. See, this all these are stamens, and the carpels are of 5 and they are united together and bifid in conditions. See, this is the stigma carpel which is fused together. They are 5 in number. One of the important character is when the flower is before pollination, the flower color will be white in color, and after the pollination the flower color changes into pink in color. See, after the pollination the color of the flower changes into pink in color. This is after pollination and this is before pollination. So for proper selection of flower is important in case of cross pollination, as the stages of the flower varies according to the growth. So proper selection of the flower is very important. So let us see some of the flowers in. So these are the different stages of flowers. So selection of the flower at right condition is very important in case of cotton. So this is the first stage, which is pinhead size stage. We can see the variation in flowers also. This is pinhead size stage, and the second one is head match stage, somewhat larger compared to pinhead stage, and the third one is square stage. As the shape indicates in the form of square, the name was given as square stage, and the third one is candle stage, and this is the most important stage where we have to select the flower for cross pollination. So we have to select the flower in candle stage in order to do emasculation and cross pollination, and the last stage is full blossom stage. So it will not, we should not select the flowers which is already blossom, because already flowers petals are all open, we can't do cross pollination. So we have to select the plant which is in candle size stage. Okay. So this is the candle stage of the flower, as the anthesis period is from morning 6 a.m. to 8 a.m. in the early morning. So now let us see self pollination. For in case of self pollination there are two methods, and in case of cross pollination the mostly followed methods are two methods. So first let us see the self pollination in case of cotton. There are two methods in case of self pollination. First one is paper cup method, and the second one is tie method. In paper cut method we have to cover the flower with candle stage flower with the paper cup entirely. So this is a paper cup, we have to cover the candle stage flower entirely, so it will prevent the cross pollination. So the flower is covered. And the second method is tying the flowers with thread. The second method is tying the flowers with thread. Only the upper portion of the flowers, which the petals which will be opened the next day, that plants are to be selected, and they should be covered tied with a thread like this. So it will prevent the outcrossing. So there will be only choice of self pollination. These are the two methods in case of self pollination in cotton. Whereas coming to cross pollination, there are two methods mainly we follow. One is docus method, which was given by scientist docus, and the second one is soda straw method. The mostly followed and easy method is docus method, because it is mostly followed, and the straw method is somewhat difficult compared to docus method. So first let us see docus method. For docus method we have to select the plant which is in candle stage of height. So let us see the flower which is in candle stage. So here is the flower is at candle stage. So slowly remove the outer sepals and make a slow cut at the end portion of the flower. Care should be taken to not to damage the ovary part. A slight cut should be made. You can make with the help of a needle or anything, but see, slightly you have to make a cut and open the petals. See, we can see the numerous number of stamens. Remove the petals totally. So this is ovary. You can see that somewhat dark in color, and these are stamens. So remove the stamens with the help of your finger, or you may use the needle also. Slightly do like this. All the stamens will be fallen. Anthers. So remove all the anthers from the flower. Only the stigma portion will be remained. So emasculation is already completed. So now in order to prevent the cross pollination, we may cover the flower with a paper bag. So all the anthers are removed. So in order to identify the flower, we have to tag the flower. The tag contains the information about the date and the type of variety already labeled here. So in order to prevent the cross pollination, we may cover the flower with paper bag, or we may use the straw, and we have to bend the upper end of the portion like this in order to prevent the cross pollination. The second method is soda straw method. Same, the process will be the same like docus method only, but in case of soda straw method, we have to take a straw and we have to insert the straw from the upper region of the anthers, and we have to twist the straw like this, rotating, so it will make to fall the anthers fall down. So let us see in another flower. So let us see soda straw method in another flower. This is also candle stage. Make a slight cut at the down portion. See, all the petals are to be removed. Only stamens are present. Remove the petals totally. Now take the straw and insert from the upper end of the stigma and twist the straw. So it makes to falls all the anthers. Only the stigma portion will be remained. See. See. All the anthers are removed. See, all the anthers are removed. Only the stigma is present. This is known as soda straw method. But mostly we follow first method, which is docus method. Thank you. Please share, like and subscribe our channel. We are students from Jagityal College. Thank you.